So my research was with Dr. Snyder, and we were working with cyanobacteria, and the main goal of this was just to determine the effects of PEG, glycerol, and fructose on the extraction and separation of phycocyanin from cyanobacteria. So just a little introduction. Cyanobacteria is a group of bacteria that are photosynthetic and typically live in very moist soils or waters. They're typically called like the blue-green algae in a way. They can produce toxins and can become very dangerous, as they are potentially dangerous, to other organisms that cross paths with them, such as aquatic organisms, farm animals that drink from ponds that have algae blooms, or wildlife that would drink from those same ponds and lakes. But what makes them so unique is the blue pigment that they all share, and that's the phycocyanin. This picture is of a dead fish in a algae bloom, so you can see how potentially dangerous it is. Alright, so phycocyanin is a blue pigment that is a light harvesting pigment. It binds proteins and is isolated from the cyanobacteria. It is widely used in food and cosmetic industries as either food colorants or like fluorescent dyes. And there's actually new possibilities and potentials for phycocyanin that's being used and studied in the medical and the biological fields. Some researchers have determined that phycocyanin can serve as an anti-inflammatory, antioxidant, anti-tumor, and while all serving that, they also improve anemia and they promote immunity. Whereas another study describes phycocyanin as a potential drug for cancer treatments. So with so much potential, scientists are trying to find and trying to determine the most efficient way of separating and extracting phycocyanin from the cyanobacteria cells. This process of separation and extraction are somewhat detrimental to the phycocyanin levels. So what that means is throughout these processes, phycocyanin is lost or damaged because it's just so extreme the way that you have to basically, I guess you could say, burst the cell membranes and get the phycocyanin out of the cells. Thus, the purpose of this research experiment is to find a substrate that will aid in protecting the phycocyanin during the separation and extraction processes. The hypothesis for this research experiment was that substrates that are very viscous, so thick, would be will better preserve the levels of phycocyanin than water would, just plain water. Alright, so I used 6 cultures of phycocyanin. I used Lemnothrix, I'm going to butcher these names, the Microcystis aeruginosus, Spirulina, Spirogyra, Declindospermium, I'm guessing, and Anchistrodinus. And then the substrates used is the PEG, which is the polyethylene glycol, glycerol, fructose, and water as my control, or like to compare. Alright, so my methods and procedures. First, I had to filter a culture. The cultures were gathered. They were filtered using Dr. J's very large vacuum apparatus. If you've been in the research lab, it's everything glass. And the VersaPore filters, which are just one of many. Once all the medium had passed through the filter, the culture was left on the filter, as you can see on the right. And when removed from the vacuum apparatus, the culture filters were actually placed in liquid nitrogen, which is new compared to my research back in the spring, for 2 minutes, and this was to freeze the cultures. The reason to freeze the cultures is because previous research has also proven that cultures that are frozen, like a freeze-thaw kind of thing, actually have better levels of phycocyanin or more phycocyanin present. It's easier to extract than room temperatures. Alright, so this was my divisions of the culture filters that are coming up. I trimmed the excess around, so it was just the culture and then it was split into equal fourths. Alright, so after the 2 minutes were up, again the filters were cut into equal fourths, and each fourth was placed into a separate microvalve containing different substrates. And each microvalve was a different compound to be tested, of course, and there was also individual blanks made, so it was the exact same thing as the one with the culture, but it was just minus the filter so you could compare them. So in the first tube, well, the microvalves are each 2 milliliters. So in the first one we did 1.25 milliliters of the PEG and 0.75 milliliters of octanol. And the octanol is used to like break the cells open. So like that was a control for everything. We used octanol in every single one of them, and we just changed the the PEG, glycerol, water, and fructose to see which one of those substrates would be the best in preserving the phycocyanin. So as you see, each one of these got a certain amount of substrate, and the 0.75 milliliters of octanol, and the 2 milliliters was total, as the total volume. Alright, so in the separation and extraction processes, once the filters were placed in the microvalves containing the substrates, it was immediately shaken using the little mini bead beaker, beater. It's a little noisy thing, for 30 seconds. This was to disturb the cells, to break them open, to get the chlorophyll and the phycocyanin out of the cells. So once the microvalves were shaken, they were placed into the centrifuge and they were spun down for 3 minutes at high speeds. The centrifuge that I used, the high speed, is actually 13.2 times 1,000 rotations per minute. When removed from the centrifuge, we had to be very careful with the tubes, as we didn't want the layers to mix back up as we just centrifuged them. So this would be, if the layers mixed, then you would have to centrifuge again. It wasn't a big deal, but you didn't want to centrifuge it again because you didn't want the phycocyanin to be more damaged. To extract the phycocyanin, a fine point pipette tip and the pipette was used. The fine tip was used to eliminate the chlorophyll and the interface debris from mixing again with your phycocyanin, as well as it was it was trying to eliminate, it didn't, because when you're eliminating or trying to extract the phycocyanin, you don't want the chlorophyll or the interface. So and the tips are like smaller than a 0.5 lead pencil, like the end of the lead pencil. And then this would be the chlorophyll, and then the interface level it's very small. And the microvalve was very easy to see as if it was a different color. That's your dead cells, your cell waste, and your filter waste, like if you had small filter particles. And then you had your phycocyanin, which of course was the blue. And then in this one, there was actually not a pellet of live cells, but if there was, it would be towards the bottom. So once a sample of phycocyanin was extracted from the microvalve, it was placed in the well plate for further analysis. So for the well plate, once all samples and blanks were placed in the well, and we used the 384 well plate, which were black with a clear bottom, because that makes a big difference when doing the well plate reader. An absorption spectrum was ran. The absorption spectrum was finished, and phycocyanin levels as well as substrate blanks were removed from the well plate, and the data collected was used to create absorption spectrum graphs for each culture. So the results, this lab suggested that the phycocyanin levels can be protected by certain vicious substances. When I say certain, that usually means there was only one that actually outdid the rest by far. Alright, so in my first culture, which was limnothrix, from left to right, this would be your PEG, your fructose, your water, and your glycerol. This is just the microvalves. It's not even the absorption spectrum, and you can clearly see that fructose had a lot of phycocyanin when it preserved compared to the other ones, with water with a close second. The PEG and the glycerol had no phycocyanin present as to the naked eye. We think this is because, me and Dr. J talked about it, and we think this is because it was almost too viscous, like too thick for the cells. Either the cells didn't lyse correctly or something like that happened. So for the absorption spectrum of this, the fructose is the orange line, and with pure phycocyanin, the absorption spectrum is going to be anywhere from like 625 to 675, and as you can see, that is a clear peak on the fructose for the limnothrix, with water being a close second. There's hardly any peak on the glycerol and the PEG, but there was no blue color in those microvalves as well. So that matched up perfectly. For MR aeruginosis, from left to right was the same. It was the PEG, fructose, water, and glycerol. Again, the fructose outdone the rest of them. There looks to be a little blue color here and a little blue color here compared to the limnothrix, but the fructose was still the winner in that one. And that's again given in this absorption spectrum, which the orange peak was fructose with the clear peak where phycocyanin should be. And then with the spirulina,